friends, welcome to my channel Biofix Buddy. If you haven't yet subscribed, please do subscribe to my channel and put an end to all your doubts. Today we'll be learning about double fertilization. So here double fertilization takes place in flowering plants, and flowering plants are also known as angiosperms. Double fertilization is a major characteristic of angiosperms. Basically, in angiosperms, reproduction takes place by the fusion of a male and female gametes. So, in case of double fertilization, which takes place in the flowering plants, there is a fusion of two male gametes with one female gamete. One female gamete unites with two male gametes. In the two male gametes, one of the male gametes fertilizes the egg cell, while the other sperm cell unites with the two polar nuclei for the formation of an endosperm. So double fertilization, it is a complex process where out of the two sperm cells, one fuses with the egg cell. So the sperm cell is a haploid cell, the egg cell is a haploid cell. Therefore, the fusion of one sperm cell and one egg cell results in the formation of a diploid zygote. While the second sperm cell fuses with two polar nuclei. So this cell, known as two polar nuclei, contains two haploid nuclei. So it is a diploid cell. This two polar nuclei is a diploid cell. It actually contains two haploid nuclei. Therefore, it is a diploid. So when a diploid cell fuses with one haploid sperm cell, it results in the formation of the triploid primary endosperm nucleus. So there is fusion. There are two fusions taking place here. That's why it is known as double fertilization. Since the endosperm is a product of a fusion of three haploid cells. So where does the three haploid cells come from, nuclei come from? Two are two haploid nuclei in the polar nuclei, and the third haploid nuclei is from the sperm cell. So there is fusion of three haploid nuclei. Therefore, it is called a triple fusion. Eventually, the primary endosperm nucleus, which has resulted from the triple fusion, develops into a primary endosperm cell. And this primary endosperm cell develops into an endosperm. So this endosperm is a component which provides essential needs for the early development of the zygote. And how does the egg cell and the polar nuclei develop is what we're going to learn now. In the female reproductive system in the flower, we have the stigma, then comes the long structure known as the style, and which ends in a swollen region known as the ovary. Inside the ovary, there is one reproductive cell which is known as the megaspore or the mother cell. This mother cell is usually a diploid, and it undergoes meiosis, which is also known as reductional division. When this diploid megaspore undergoes meiosis, it results in four haploid megaspores. Four haploid. Out of these four haploid megaspores, three of them degenerate, and the one remaining megaspore, there is one megaspore remaining in the ovary now. That one megaspore expands and becomes big, and it undergoes three rounds of mitosis. At the end of three rounds of mitosis, there are eight haploid nuclei, which means the one haploid nucleus divides into two. The two divides and it forms four haploid nuclei, and the four haploid nuclei divide and it results in eight haploid nuclei. In this process of mitosis, there is no cytokinesis. That is why all the eight haploid nuclei are present in a common cytoplasm. So this structure, which contains the eight haploid nuclei in the common cytoplasm, is now known as the embryo cell. So all these eight haploid nuclei form separate cell walls now, their own separate cell wall. Out of these eight haploid nuclei, three of reach the opposite region of the micropyle. Here, the micropyle is an opening through which the pollen grains enter into the embryo sac. So, at the opposite end of the micropyle, three of them are known as antipodal cells. Just below the at the region of micropyle, there are three cells, two of which are known as synergids, and one is known as the egg cell, to which one of the sperm cells fuse with. And now we have seen three over here, three over here, therefore six. And the two remaining haploid nuclei are located centrally in the embryo sac. So these two haploid nuclei form a common cell wall. So here there are two polar nuclei that's inside a common cell wall. That's why it's called a two polar nuclei. And the other cell, sperm cell comes and fuses with this two polar nuclei at the center of the embryo sac. And this two polar nuclei and the sperm cell fusion develops into the endosperm, which helps in the early development of the zygote, which is the result of the egg cell and the sperm cell. Here we have learned what double fertilization and how does it take place. So here we have the pollen grains. During the fertilization, the pollen grains reach the stigma. Through the style, pollen grains develop a long tube-like structure which reaches the micropyle structure. The sperm cells enter into the embryo sac. After they enter into the embryo sac through the small opening called the micropyle, they fuse with the one of the two sperms fuse with the egg cell, the other one fuses with the polar nuclei. The one which fuses with the egg cell develops into a diploid nucleus. The other one which fuses with the two polar nuclei develops into the endosperm. So we have learned about the process and what double fertilization is. Just let's just have a summary. Here, double fertilization is a phenomenon which takes place in the angiosperms. It is a major characteristic of the angiosperms. In this process, one female gamete unites with two male gametes. So in these two male gametes, one of the male gamete fuses with an haploid egg cell, which results in the formation of a diploid zygote, while the other sperm cell fuses with a two polar nuclei, which contains two haploid nuclei, which results in a triploid primary endosperm nucleus. Since the endosperm is a product of a fusion of three haploid nucleus, that is a haploid sperm cell and the two polar nuclei which contains two haploid nuclei. Therefore, it is called as triple fusion. So eventually, when there after the fusion of the three haploid nuclei, the primary endosperm nucleus develops into a primary endosperm cell, and at the end it develops into an endosperm in the early development of the zygote. So this is what the female reproductive structure looks like: the stigma, style, and the ovary. When the pollen grains reach the stigma, they form a long tube-like structure inside the style through which the two sperm cells enter into the ovary through a small opening known as micropyle. Thank you and hope you have understood the concept.